Everyone has to click the got it there. First of all, I want to thank our extension partners. Without their support, we would not be able to bring you programs like these throughout the year, and especially during the last 2 years. So thank you very much to all of our extension partners that are shown on this slide. All right, next, if you'd like to ask a question live, there's a couple ways you can do that. You can type the question into the chat box, or you can raise your hand in Zoom by clicking on the reactions button on your screen, and we will call on you to ask a question. Remember to unmute so we can hear you. And also, closed captioning is enabled, and you can turn it on and off with the button marked CC or live transcript, as is shown here on the slide. We will have let me go to the next slide here. First of all, thank you all for attending. We will have a survey that Caroline will put in the chat box at the end. So please give us your feedback. It helps us to provide relevant content. And then a recording of today's program will be emailed to those of you who are registered within 48 hours of the program. So.
I think it's my turn.
Well, hold on, I'm going to stop sharing my slide. However, I want to thank our speaker, Megan Bartlett, for presenting and answering questions today. Thank you to Caroline for taking care of the Zoom logistics. And thank you to all of you for joining us today. So now, without further delay, I'm going to turn it over to both David Block, who is the department chair and also professor in the Department of Viticulture and Enology, and to Anita Oberholster, who's an associate professor of cooperative extension and now one of our new vice chairs in the Department of Viticulture and Enology. And she is going to introduce today's speaker. Take it away, Anita.
Sorry, jumped the gun. That's so me. So it's my pleasure to introduce our speaker today. But firstly, I want to welcome everybody that zoomed in. You know, because we couldn't really comfortably do on-campus events in the last 3 months, we decided it was high time to do another office hours. And obviously you guys agree because here you are. So welcome everybody. We asked Megan to talk about the work she does because she does some really relevant, interesting research at the department. And she has a very interesting bio. So I hope I'm going to surprise some of you. So Megan did her undergraduate degree at Harvard University. Wow. And then she went on and did her PhD. In 2016, she completed a PhD with Dr. Lawrence Sachs at the Department of Ecology and Evolutionary Biology at UCLA. And her PhD work identified traits that are crucial to a whole plant drought tolerance, right? So she has been doing this work for a while. And then Dr. Bartlett went and did a postdoctoral postdoc, I should say, work with Dr. Stefan Pacala in the Department of Ecology and Evolutionary Biology at Princeton University. And there she applied optimization theory from economics to plants to understand how plants should adjust their gas exchange during drought to maximize their long-term performance. So pretty cool stuff. And so we're very grateful that she joined the Department of Viticulture and Enology at UC Davis in the winter of 2019. So she is one of the newbies still, and it may be 5 years before we stop referring to you as such. So be warned. I think it took about 7 years for me. So her research uses modeling and experimental approaches to address fundamental questions about the physiology, traits, and mechanisms that produce drought and heat tolerance. And I'm going to say no more because she's going to say the rest in her own voice and much better than me because I did get tongue-tied. I thought it was going to happen. So welcome, Megan, and over to you.
Thank you so much for that nice introduction. Thank you so much for having me today and to everyone for coming. I'm going to get my screen up. Sorry, got to move some Zoom windows around. All right, can everyone see that okay? Okay, great. So yes, as Anita said, today I'm going to be talking about the work I've been doing for the last few years to identify traits that are important to drought tolerance in grape rootstocks. And the goal of this work is to identify traits and develop rapid phenotyping mechanisms, methods rather, that we can use to target these traits in breeding for more drought-tolerant rootstocks. So drought-tolerant rootstocks are a really important management tool to maintain canopy growth and yield under dry conditions. And that also helps maintain a suitable microenvironment for the fruit and berry quality. So California is naturally a system that experiences periodic droughts and heat waves, and these are projected to become more intense in the future. So identifying strategies to improve the drought tolerance of grape rootstocks is a really critical priority for industry. So the goal of this work was to identify traits that confer drought resistance in grape rootstocks so that we can amplify these traits through breeding. And so in this work, we really wanted to zoom in on specific traits that we expect to be especially important to drought responses. And so the roots of a grapevine are a really important bottleneck for water transport of the whole vine, especially under water stress. So even under well-watered conditions, about 90% of the resistance to water movement from the soil to the canopy comes directly from the root system. And this is even more exacerbated as the root system dries down. And so most of that resistance to water movement actually comes from a very specific tissue within the roots. And so it comes from a very narrow band of living cells in between the epidermis at the boundary of the soil, and then the xylem, that's the specialized water transport tissue to bring water up to the canopy. So this is an example of this band of living cells. So all water that moves up to the canopy has to cross through this narrow band of cells. And a lot of evidence coming from really cutting edge imaging techniques is suggesting that this is really the most crucial bottleneck, that these living cells are the things that cause most of the resistance to water movement under drought. And so a couple of things can happen to those tissues under drought. So the first is that they can actually just physically shrink as they become dehydrated. And so that process of shrinking actually causes the roots to become disconnected from the soil. So this is an example. This is a cross section through a root in a soil. And so you can see that the actual cylinder of the root has pulled away from the soil and it can no longer access the soil water. The second thing that happens is actually work that was discovered by Dr. McElroan in our department, which is that in grapevines, as the roots dry down, they can actually experience a collapse and a death in these cells, in these normally living tissues. So this picture is an example of that, where this is a well-watered root, this is a droughted root, and all of the areas that are dark in this image are areas where these cells have died and they've become kind of air-filled voids. And so having that cell death actually interferes with the ability of the root to move water because you're losing pathways for water to move. So that's an example here. You can see as these cells drop out, there are fewer pathways for that water to move from the soil to the xylem. And so what we wanted to do in this study is to identify traits that can capture these responses in the living tissues. And so I'm going to zoom in on two that actually turned out to be important in this study. And so the first trait we focused on is called capacitance. And so that's measuring the relationship between the volume and the water potential inside the root. So you can think of it as a rate of root shrinkage as the water potential declines. So in this case, a root that has a very large capacitance would undergo a lot of physical shrinkage under water stress. And so that would be a root we would expect to be more sensitive to drought. It would become decoupled from the soil earlier. The second trait that we focused on is called the turgor loss point of the root. And so the force that holds up the inside of every cell in a plant is called turgor pressure. And so this is the water pressure of water pushing out against the inside of the cell walls. And so as cells lose turgor, they lose this force that holds up the cells, and that causes the cells to collapse and the tissues to collapse as well. So you can see this in the leaves. When you see a leaf wilting, you've seen that leaf become so dry, there's no longer enough water to create this turgor that holds up the cells collapse, and that collapses the whole leaf with it. And so a similar process can happen in the roots. And so our expectation was that this turgor loss point would tell us about this process of these cells collapsing and dying in the roots. So we expected that a more negative turgor loss point would indicate more to be beneficial to the structure of the root by avoiding collapse, and also the growth of the root, because this turgor pressure is the driving force for new cells to expand in growth. So those are the two traits I'm going to focus on today. And so I'm going to talk about a series of 3 experiments that we've been doing over the last few years to look at these traits and their connection to vine drought tolerance. So first, we wanted to get an idea of do rootstocks actually vary in these traits, our common commercial rootstocks? And also, do we see whether or not these trait values are actually impacted by drought? Can a plant that's exposed to drought actually kind of alter its anatomy and structure in a way that makes these traits more suitable to drought? Second, we wanted to look at, are they actually related to the aspects of vine performance that we're interested in under water stress? Do we see that these traits improve the gas exchange and the growth of the scion under water stress? In our newer experiments, we wanted to get a better idea of why these traits work, what mechanisms actually connect them to drought tolerance. For example, how do they affect water transport under drought? And then finally, we wanted to look at these trait values across different experiments to see, are they consistent and predictable across rootstocks, which would be a necessary condition for breeding. If we screen for these over and over again, do we actually see repeatable values? So I'm just going to go through the 3 experiments really briefly. There are 3 greenhouse drought experiments. So the first one involved 8 rootstocks grafted onto Chardonnay, and they were exposed to 2 watering treatments, where half the plants were kept well watered and half the plants were kept water stressed for 3 months. And so this long period allowed the plants to acclimate to drought. So we sampled plants to measure these root traits, and the plants that we weren't sampling for the root traits, we were monitoring for gas exchange, water stress, and then at the end of the experiment, we measured the biomass above and below ground. The other 2 experiments we conducted over the last summer. So here we zoomed in on a couple of these rootstocks that were especially different in these traits. And we wanted to subject these rootstocks to rapid droughts because we wanted to understand what these traits mean with and without a long acclimation period. For example, in a vineyard, kind of what's happening when your vines that haven't been water stressed are suddenly experiencing a period of water stress. We wanted to see what these trait values mean for water uptake from the soil. And then finally, we repeated this experiment with the 4 of the rootstocks from the first experiment grafted onto a Cabernet Sauvignon. And we wanted to see how repeatable these trait measurements are. Are these going to be reliable traits for selection? Okay, so what did we find? So first, we found that these traits were very diverse among commercial rootstocks. So these are our 8 commercial rootstocks. 4 we would expect to be sensitive to drought in the field. 4 we would expect to be tolerant to drought. And so the blue bars here are showing the well-watered plants. The gray bars are showing the water-stressed plants. And so just looking at the turgor loss point and the capacitance, there's about a 3-fold variation across all of these different rootstocks, just within the well-watered treatment for both of these traits. So there's a substantial amount of variation already in the rootstocks we already use. Another thing we found is that these rootstocks actually adjust in a way that you would expect to improve drought tolerance under a prolonged water stress. So you can see here, the turgor loss point became more negative in the plants that were allowed to acclimate to a long drought. So we would expect these roots to be more resistant to this cell collapse and cell death. And capacitance also became smaller in the roots that were allowed to acclimate to drought. So we would expect those roots to have more structural integrity and maintain more connectivity with the soil. So this was promising. There's a good amount of diversity in what we already use. We see that grapevines already naturally have an ability to acclimate these traits. So what do they actually mean for the drought tolerance of a whole vine? And so what we found is that the capacitance was really critical to the gas exchange of the scion under drought stress. So this is showing stomatal conductance related to the root turgor loss point and the root capacitance. Each point is the average gas exchange for the experiment. The black is the droughted vines and the blue is the well-watered. So we didn't find a relationship between the root turgor loss point and the stomatal conductance, but we did find a significant relationship with the capacitance, where the roots that had a higher capacitance, which we would expect to mean more shrinkage away from the soil, were less able to keep their stomata open and maintain gas exchange under water stress. While the rootstocks that had more structural integrity and did less shrinkage were able to keep up this gas exchange and keep up stomatal conductance. And we saw similar results for photosynthesis. So that went along with our expectation that this trait is related to structural integrity and the ability to take up water from the soil. For turgor loss point, we did find a kind of a weak connection between the size of the root system, which we kind of were expecting from the relationship between turgor and growth, and a very weak relationship with transpiration of the whole plant. So this is looking at the relationship between turgor loss point and the root area. So there's a very weak relationship where the rootstocks with a more negative turgor loss point tended to have a larger root area under drought, which we would expect since this trait is related to the ability to maintain turgor, and turgor is what powers growth. And we saw a very weak relationship between turgor loss point and the whole plant transpiration under drought, but for the water stress plants, they all basically converged on the same level of whole plant transpiration. So there's a little bit of a role for turgor loss point in growth, and there seems to be a good role for capacitance in gas exchange. And so our second experiment was trying to look at what does capacitance actually mean? Do we actually see proof that capacitance is helping to keep up this water transport? So in this experiment, we zoomed in on a rootstock that is generally high capacitance, a rootstock that's generally low capacitance. So that's 420A is high, 140 rupestris is low. They converge to being fairly similar under acclimated, when they acclimate to stress. So we subjected them to an experiment where some of the plants we kept well watered and then subjected to a rapid dry down. So we wanted to take away their time to acclimate and understand what these traits mean when they're exposed to a sudden drought. And then half the plants we allowed to dry down, to kind of sit at a consistent level of drought and acclimate to that, and then dry them down further to see, do we actually find evidence that acclimation is helping these plants improve their drought tolerance? So what we found is that so this is the conductivity of the soil to the plant. So this is measuring the amount of water transport divided by the difference in water potential between the soil and the plant. So generally a very high conductivity indicates a plant that is highly capable of transporting water. And this is plotted against the pot weights. So as the pot weight percentage is getting smaller, the plants are drying out. So the red points are showing the high capacitance rootstock, and the other points are showing the low capacitance rootstock. So for the plants that weren't allowed to acclimate, where capacitance was very different, we actually low capacitance is helping the plant maintain water uptake. Where there is these relationships were significantly different between conductivity and the dryness of the plant. And the 140 RU is maintaining more of this water transport and water uptake for a given level of drought stress. We didn't find in this experiment a benefit from acclimation. We didn't find that allowing the plants to acclimate for a period improved their ability to maintain conductivity. In this case, it might have been an overly short acclimation period. Here it was a couple of weeks versus a few months in our previous experiment. But it kind of raises the question of, what does this acclimation mean? Are we really measuring the growth of new roots that are designed to be better at drought performance? Or are we kind of measuring the tissues that are left over after a root has been stressed for a long time and all the vulnerable tissues have died off? So we're kind of doing more work to understand what this acclimation period really means. And then finally, we found that the turgor loss point was very well correlated between experiments with different scions, but we didn't find that with the capacitance. So I have a little bit more of the results from that to show you a little bit later on. But generally, this kind of is a knock against capacitance as a good trait for breeding because it suggests it's very sensitive to the growing conditions or the scion. But turgor loss points seem to be something that was reliable and potentially could be selected for. Okay, so overall, we found that the root cellular traits seem to be important to rootstock drought tolerance, where we found that capacitance and turgor loss point are correlated with vine gas exchange and root biomass under water stress. We found evidence that the roots with a lower capacitance maintain better connectivity with the soil for water transport. And so that suggests that those rootstocks have a greater capacity to extract water from the soil. That as the soil gets drier, they're still able to keep up their water uptake from that soil. The rootstocks with a lower turgor loss point produce larger root systems under drought. So that suggests that those rootstocks have a greater capacity for soil exploration. They're able to keep moving into new undepleted areas of the soil. And then finally, we found that turgor loss point was very predictable, but capacitance may be a bit too sensitive to growing conditions to be an effective target for breeding. So this increased extraction capacity from capacitance is interesting though, because it could potentially be important during fruit development. So very little new root growth takes place after veraison in order to avoid resource competition with the fruit. And so that's what these results are showing here, that very little of the root growth over the life cycle of the grapevine actually takes place from veraison to harvest, which is often the hottest and driest period of the growing season. So that suggests that the exploration of new soil is limited during this time, and the ability to extract more soil where you already have roots could potentially be really important in that period. Another way that capacitance could potentially affect drought tolerance is that the actual physical shrinkage of the roots actually causes a hormone production called abscisic acid or ABA. And ABA has implications throughout the plant, where one hypothesis is that it's a signal for stomatal closure. It's transported up to the canopy and causes the stomata to close, which is potentially another explanation for our relationship with capacitance and stomatal conductance. But ABA is also a really important signal for ripening, especially for the onset of veraison. And we've seen that scion cultivars differ in their sensitivity to ABA. So this potentially suggests that if this is the mechanism by how capacitance works, we need to think about what breeding for a low capacitance rootstock means. Is it effective for every scion? Does it have potential downstream effects on fruit quality and ripening? So there's been some really interesting work in other species that kind of raises questions about whether this ABA mechanism is true, even though this has been kind of the dominant paradigm in grapevines for a long time. So there's some really interesting questions there. Is the signal be something else, or does this signaling even happen at all? So it's a really interesting area of kind of what do these traits mean with hormones and ripening. So turgor loss point is also interesting because it's potentially important to root growth and lifespan, where we know turgor loss point improves root growth under water stress for many species, but turgor loss point is also potentially important to root survival in dry areas of the soil, wherein grapevines are very effective at moving water from wet areas of the root zone to dry areas of the root system through a process called hydraulic redistribution. And it's been shown that that process helps to keep these roots at the surface alive. And it's always been assumed that this is beneficial, but root respiration is expensive. Keeping roots alive is very expensive in terms of carbon. That can account for about 70% of your photosynthesis during drought. So that kind of raises some questions about, is more always more for these traits? Or do we want is there kind of an intermediate value that's optimum that would allow the vine to sort of kill off roots that are in very dry soil and very depleted soil in order to redirect resources to growth in wetter soil? What is it what values would actually be most beneficial for breeding? And then finally, is it really true that turgor loss point is a better candidate for breeding than capacitance? So what we found by comparing the capacitance to the level of drought in our different experiments was that our first experiment was very homogeneous. That's these green points here. So within each treatment, we were very good at narrowing in on a very narrow range of pre-dawn water potentials. So kind of all the plants in each treatment were experiencing the same level of stress. We were not as good at that in the second experiment, because in the first experiment, we were using relatively large plants in relatively small pots. So in our second experiment, we're potentially seeing rooting depth have more of an effect, where the tolerant rootstocks have their roots lower in the pot, and so they're experiencing less water stress. Their capacitance is higher, even though all these plants are getting the same amount of water or level of stress. So that suggests that effectively screening for capacitance is going to require a very careful control on growing conditions. So that raises questions about how well can we actually target this trait in breeding, and what would an effective screening mechanism for this trait really look like? And under what conditions is capacitance consistent across soil types and scions? Okay, so that we've been doing is thinking about kind of how to take this work and make it effective for breeding for rootstocks that are going to be useful in a vineyard setting. So we're interested in what range of capacitance and turgor loss point values are actually most beneficial to drought performance. Is this kind of a monotonic relationship where lower is always better, or is there some kind of intermediate value? And how does this value depend on the scion, cultivar, or the soil type that we're in? That ABA is a potential way of thinking about the effects on the scion cultivar. Are we potentially going to have effects on ripening independent of the level of drought stress? We're interested in how would you actually produce a root system that has a low capacitance or turgor loss point? So what anatomical or biochemical characteristics would you expect to see in these roots? And then finally, we were thinking about these as pots, plants in a pot where the roots are either water stressed or they're well watered. But within a vineyard, grapevines are very deeply rooted. So they're probably experiencing, they certainly are experiencing dry and wet soil at the same time in most conditions. And so what we want to know is, in an actual grapevine in the field, do we see a combination of different trait values? Where are the traits we measured for water stressed conditions present in the surface, while the traits we measured in well-watered conditions present deeper in the soil? So that kind of raises questions about what does that variation within a grapevine mean? How can we kind of manipulate that to make a grapevine that's really effective at taking up water from these more reliable, deeper root zones? So with that, I'm really happy to take questions. I want to thank everyone who supported this work. This was a collaboration with the McElroan and the Walker Labs. We've had a lot of students working on this project, undergrad and grad students. This work was supported by the American Vineyard Foundation. Novavine donated some of the plants for this project, and also just a lot of support from the department and the college. So thank you so much for coming. If at any point you would like to talk more, this is my email, you have it, so just let me know.
Great, thanks, Megan. Just a reminder to everybody on the line, if you have questions, you're welcome to put them into chat, or to raise your hand and we'll call on you to unmute and ask the question. But let me kind of start off with a question. So you talked a lot about the characteristics of roots that are desirable for drought. So let's say we're pretty sure that drought's going to continue being an issue in California, which it's likely to be. So what would an ideal root look like in a rootstock?
That's such a great question. So I think that we want to keep up the traits we already know are effective in rootstocks, which is closely related to a deep rooting depth. We want to keep up that phenotype, like 110R and 140RU have, that they're very good at sending roots deeper in the soil. And I think we also want to work on this extraction capacity. I think we maybe want to target being able to keep up root growth under mild stress through traits like the turgor loss point, making turgor loss point more negative. So as the grapevine is depleting the soil around it, it's able to continue that growth. I think that will be really effective with helping with this deeper rooting phenotype. And I think also increasing the extraction capacity through capacitance would be beneficial, where being able to kind of stay in contact with that drying soil and to keep moving in water, I think would be especially effective during veraison when root growth is limited because all the resources are going towards the fruit. And I think also kind of the future work will involve kind of understanding how these traits can kind of be interlaid with other aspects of resistance, like disease resistance or excluding salt or improving nutrient use efficiency. So I think that the kind of stacking those traits into the optimal rootstock will be really important.
Well, that's very related to a question that Kevin put into chat. The question is, you once had a session about Pierce's disease tolerant rootstock. And are there, I'm guessing the question is, are there rootstocks that are both drought and Pierce's disease tolerant, and what changes in flavors do they create, if any? Kind of the idea of stacking these traits, right?
Yeah, so drought and Pierce's disease resistant. So there's believed to be kind of a connection between the xylem anatomy in both Pierce's disease and drought responses. Whereas the xylem is actually what I didn't talk about much today, but that's like the specialized water transport tissue that's kind of acts as pipes to bring water up to the canopy. Today I was kind of talking about that first step of going from soil to xylem. But it's believed that having larger xylem makes you more susceptible to drought and also more susceptible to the spread of Pierce's disease, probably just because you have a collection of fewer large pipes versus a lot of smaller pipes. So any damage, anything that knocks out some of those pipes you don't have that much redundancy in the system to continue your water transport. So that can be knocked out by either drought-induced damage or Pierce's disease-induced damage. So through that relationship, it's believed that having a xylem with a lot of small conduits is potentially beneficial to both Pierce's disease and drought resistance. So in terms of flavor, I think for the most part, under viticultural conditions, the rootstock doesn't necessarily have a lot of impact on flavor. It's mainly yield and vine balance in most situations. Like if you start if you're very drought stressed, you start having severe problems with sunburn and things like that, then having a large enough canopy to prevent those kinds of issues becomes a quality issue, and that's related to the rootstock. But most of the time, if you've got adequate vine balance, you're not seeing that huge of an effect of flavor from the rootstock.
Another question came in through the chat,
Okay,
Megan,
great.
so I'll read it for you. We used to understand that wine quality was positively impacted by the ability to provide early season water deficits to the vines. Have we seen any research that suggests increased drought tolerance will alter that understanding?
No, I don't think that's, yeah. So I think that what most people are picturing is that to get the optimal levels of stress in the future, the levels that are optimal now to reach those again in the future, we're going to need either more irrigation or more capacity to bring up water from the soil to reach those same levels of stress. Sorry, I think I'm not explaining that very clearly. I think in the future, conditions are expected to be hotter and drier while fruit ripening is happening. So in order to reach those same water potentials that we want to target for optimal ripening, we're either going to need to apply more water or we're going to need a rootstock that's better at reaching water that's in the soil. So I think that's how most people are thinking about it. It's like, we need that rootstock that's better at getting water and bringing it up to stay in that same range of water potentials. Not so much that we've fundamentally altered how water stressed we want the plant to be for ripening. But it's a really interesting question. Like, I think there's a lot about rootstock to scion signaling that we don't necessarily know. And I think that'll be something that's really important to kind of get at in breeding. It's like, what are the implications of ripening coming from having a different communication between the rootstock and the scion?
So there's a question in the chat that's a follow-up on what you were saying. I think about xylem size and whether it's better to have one large xylem or smaller number of large xylem or more smaller xylem. And the question is, could the same theory, RE xylem size, apply to trunk disease susceptibility?
Yeah, so that's another great question. So I think that yeah. I'm thinking about that because the Pierce's disease tends to move through the xylem directly, while like trunk diseases tend to just kind of destroy, like they just bulldoze through the whole stem tissue. Like they're just kind of destroying everything in their wake. So they're like a little bit less dependent on like the actual structure of the xylem, I think. So I would imagine there probably is a role there, but I would imagine maybe it's a little bit less than Pierce's disease, just because like they're just doing so much broad spectrum destruction.
Okay, so I have a question. So what kind of work are you doing to make these traits, the capacitance and turgor levels, useful for breeders? Meaning basically that connection between the basic research you're doing now and how people can actually use that information to apply it in the field.
Yeah. So one of the things we're trying to develop some rapid phenotyping methods for these traits, because they're very time consuming to measure in this greenhouse assay. And so one of the things we're experimenting with is taking roots and just putting different concentrations of solutions and photographing them to measure how much the roots are shrinking and to try to automate that process, with the idea being like, oh, if you were looking across a mapping population for breeding, you could just take samples of roots and put them in these solutions and photograph them over time. So that's one of the things we're doing is trying to actually like speed up this phenotyping. The other thing we're doing is we're starting some new work where we're actually trying to look for genes that are related to rootstock drought tolerance traits. So these are some of the traits we're interested in. We're also interested in traits that are related to rooting depth and rooting distribution. We're looking at a population that's a cross between 110R and 10114 as a tolerant and a sensitive cross. So we're hoping that will enable us to pick out some of the genes that are related to these traits, so we can actually then really speed up breeding by having molecular markers that you can go through. That's been one of the things that's kind of held back breeding for abiotic stress tolerance, is because we don't have those kinds of genetic markers the way Andy Walker was creating for disease resistance. So I'm hoping with that work, we can actually start to identify these genes and really speed up that process.
So I have a question for you. So you I guess, if you think about evolution, these vines that we're using today, whether it's rootstock or scion, have evolved over time or been selected for certain traits. And I'm wondering, so if we get a rootstock or scion that's very drought tolerant, what are we going to lose? Is there some trait that we're going to lose that's going to make it not as good as what we have now? I realize there may not be a choice in order to have viticulture in a drought prone area. We may have to go that route, but what might we lose?
Yeah, that's such a great question. So So when we go to like the wild species that are the parental varieties for a lot of these rootstocks, a lot of them grow kind of in like riparian areas, like they're right on the banks of like a river, like so they don't really experience like drought stress where they are for the most part. So their evolutionary history like hasn't really been one of dealing with stress. Like basically like they have evolved to try to get up in a tree and dominate that canopy of that tree as quickly as possible. So I think by selecting for like deeper rooting, a more extensive root system, maybe more structural integrity in the roots, I think we would potentially be directing more resources towards root growth early on, which I think would be a major loss for that wild grapevine, because I think that would slow down that initial push of growth into the canopy. And in a viticultural sense, like maybe that is potentially like slowing down the establishment of young vines, maybe, but I think in a lot of cases, like building more root growth actually builds more, speeding up, or that's not exactly the right word, allocating more resources to root growth, like is like a virtuous cycle. It's a positive feedback, like not a trade-off, because you've got more resources to support photosynthesis. You get a larger canopy, you get more resources coming in for both canopy and root growth. So I'm actually not totally convinced we would see a trade-off in a viticultural sense. Oh, I'm sorry. I think you're muted.
I am. Sorry about that. Maybe I'm thinking about this too simplistically as a chemical engineer, but you know, you hear anecdotally people, you take tours of wineries and stuff, people always talk about, you know, they need their grapevines to struggle to get good quality fruit.
Yes.
And if you now let's say, make it easier for the vine to survive, would you expect that to affect the flavor in some way?
Well, so I think the expectation is that the struggling is going to come from the environment, where we're going to be putting these vines in a harsher environment, and to get the same level of struggle, we need to actually help them. So that's actually I think maybe that's a better answer to a question somebody asked in the chat. That's probably the more concise, succinct answer to that. They need a little bit of help to have that same level of stress, is the expectation. And so, you know, like these rootstocks may be really important for some areas, like the Central Valley, areas that we expect to be hotter and drier, or potentially for irrigation to be more limited by kind of legal and environmental regulations. We may not need these supercharged rootstocks everywhere. Like maybe in some places where we're currently using devigorating rootstocks, maybe we use rootstocks that we already have that are less devigorating. So I see these as kind of like pushing that envelope a little farther to help these places where they're going to be struggling too much for profitable viticulture.
Okay, Megan, so now I'm going to ask a question, and hopefully I understood your presentation well enough that this comes out right. So from what I remember from the graph that you showed, so lower capacitance and lower turgor in general seems to indicate better drought tolerance, right, for a rootstock. Yeah. And you did a screening of 8 rootstocks, 4 that's supposed to be known as more tolerant than others. And there wasn't 100% correlation, right, from what I saw. And I'm just trying to think about, are there potentially other traits that you're not looking at that could be, with this, a better indicator of drought tolerance? And how did they in the past decide which rootstocks are drought tolerant or not? I'm just wondering, what did they test? You know, how was that done?
Yeah, that's a great question. So we chose a greenhouse experiment specifically to get rid of the effective rooting depth, because we know that's important. That having, being able to grow deeper into the soil, we already know is a really important characteristic of a high vigor and a drought tolerant rootstock. And by drought tolerant there, I mean ones where you go into the field, you put the same scion on all these different rootstocks. in the same site, and you give it low irrigation, and you see a larger canopy and a higher yield. So that's what drought tolerant means. That's where these classifications of drought sensitive and tolerant rootstocks have come from. And so we deliberately wanted to kind of get rid of that to understand what traits can we stack on top of rooting depth, so we can kind of build on that knowledge. That's kind of been, that's been like well established. So that's kind of where like, so I've got a question in chat. So sorry, let me finish what I was saying there. So so there's not 100% correlation because we've eliminated that effective rooting depth. And I think if we improved like both this rooting depth, this exploratory capacity, and we improved this extraction capacity through capacitance, that would be like kind of two drought tolerant traits stacked on top of each other.
Yeah, basically what you're doing is adding on to what people already have been evaluating.
Yes.
So no, that makes sense. It's not totally field applicable just as it is.
Yes, not yet. Yeah.
Not yet. So I'll ask this question in the chat, just because it's a sort of a follow-up on what I made you talk about by asking my question. So how do you define drought tolerance for grapevines? And what's the relationship between drought tolerance and yield in grapevines?
Yeah, so what I would say there is that from a viticultural perspective, what you want is a plant that maintains its function under low irrigation or dry conditions. So for us, that's going to be a plant that maintains enough canopy growth and maintains enough yield. So I think by definition, like yield is going to be a part of drought tolerance. I think this is not necessarily I think I'll stop before I talk in circles and become confusing. But yeah, like for viticulture, I think a plant that keeps on being productive with low irrigation or in dry conditions is what you want.
So we have about 10 minutes left. And before we go back to questions for Megan, I just want to, first of all, again, thank Karen and Caroline for organizing today's office hours. And to remind everybody that actually a week from today at the same time, so on the 17th, Thursday the 17th from 1 to 2, we'll have another office hours. We'll be featuring Professor Ron Runnebaum in our department to talk about sustainable winemaking. And we'll also have special guest Nate Weiss from Silver Oak, who's in Silver Oak has spent a lot of time thinking about sustainable winemaking as we have with their 2 LEED Platinum certified wineries in Napa and in Sonoma. So we're very much looking forward to that. Caroline's dropped the registration link in the chat if you'd like to register for that. And so again, thank you to the organizers, and I hope you'll come and register for next week's. And of course, I want to thank Megan for being our guest this week as well. So going back to questions, you talked on one of your first slides, you showed the roots going through the soil, and obviously in the soil there's more than just the kind of brown blob that you have in your picture. You're going to have things like microbes and so forth, the whole soil microbiome. And I was wondering if you could comment on how the soil microbiome could affect these traits that you're measuring. Yeah, so right now we don't know at all. So for example, arbuscular mycorrhizal fungi can really improve the connectivity between the roots and the soil. And allow the roots to access a much larger zone of soil than there are actually physically roots in. So it's potentially what we're measuring is just capacitance as a way of getting at the connectivity between the root and the soil could be completely changed if there's a really extensive mycorrhizal fungi connecting that root to the soil. And we don't know, is that the case? Could the root be shrinking, but the mycorrhizae is still connected? Does it matter what kinds of mycorrhizae? Right now that's a totally open question of kind of that interplay between the structural integrity of the root and the mycorrhizae.
yeah, so I think that's still really an open question.
Okay, so these traits, capacitance and turgor, okay, what about their interaction with different management strategies that people may be employing in their vineyards?
Yeah, so I think it's interesting to think about how they might interact with irrigation, for example, because if it is true that in a deeply rooted root system, you would have like a diversity of these traits between the surface and the well-watered soil, I think that raises the question of like, what do these trait values mean at the surface that's different than what they mean at depth? And how does that interact with how frequently irrigation is coming into the system? Like, for example, like, would we want roots to stay connected to this dry soil at the surface if we're irrigating, once a week or, once every few weeks? Like, is there still a benefit to kind of maintaining this connectivity with the soil at the surface if we're doing irrigation less frequently? Would it be better to kind of kill off those roots and direct all of our resources to deeper root growth? That might actually, we might want to have a less negative turgor loss point or a smaller or a higher capacitance at the surface to help with that. Or if we're in a system where it makes sense to irrigate more frequently, then it would potentially make sense to have this active, living, well-connected root structure up at the surface. So those roots can really rapidly take advantage of that irrigation as it comes in, instead of kind of waiting for it to percolate to deeper layers. So I think there is a potential interaction there between kind of your irrigation timing, your irrigation amount, and what traits you would want and how you would want them to be distributed through this root system. So I think that's going to be something that's going to be really interesting to think about when we think about what these traits mean in a vineyard versus what do they mean in a pot.
Okay. Do you think canopy management, like the amount, the canopy could have an impact? I'm just wondering, it may be just artificial, but part of drought tolerance, like you said, was the amount of water that they get and how big their canopy grow. But if you keep your canopy small for various reasons, does that change that ratio at all?
Yeah, that's a great point. So, I mean, I think at that point, if you were like, well, my goal is to keep my canopy small, like probably you wouldn't want necessarily the most drought tolerant rootstock. You might want something that's a little bit more devigorating. But yeah, that's a different distribution of rooting depth and potentially these traits. So yeah, I think that's a really interesting point too.
So I actually have a couple more questions in the chat, and we have about 5 minutes left. So there's a question, which would be more sensitive to higher vine density, capacitance or turgor pressure?
Yeah, right now I don't know. Like both of these traits seem to be sensitive to the level of water stress experienced by the plant. The plant seems to change them under water stress. But how that would play out in a vineyard based on vine density, I'm not sure yet. It's a great question. I think we'll have to kind of just see that.
Another question in the chat is, what about the effect of variety sign on root growth, capacitance, and turgor pressure?
Yeah, right now, we don't know how much the scion is affecting that. So we did see a difference in capacitance between the rootstocks that were grown on Chardonnay versus Cabernet. Those were very uncorrelated. It's not clear like whether how much of that was scion versus just growing conditions. We had kind of slightly different levels of drought, slightly different lengths of these experiments. But I think there's definitely a potential because the scion controls like how much resources the root gets for the most part. So I think it'll be really interesting to kind of see like, does this level of resource allocation like change what these traits end up being?
So there's another question in the chat. What's the impact of cover crops on reducing water needs?
Right. So like, I guess that can cut two ways where, you know, cover crops can reduce your evaporation and kind of help keep water in the soil compared to evaporation from bare soil. Mowing the cover crop under can increase the organic matter in the soil, which helps increase the water holding capacity of the soil. You can also have cover crops that are competing with the vines for water, so they can actually reduce the amount of water available to the vines. So I think it kind of depends on what you've planted and kind of when you're letting that cover crop be active.
So given what you know right now, which obviously we don't know everything about this, given what you know right now, thinking about if you were establishing a new vineyard and you're going to go plant rootstock, aside from the choice of rootstock, let's say you pick a real drought tolerant one, is there something that you can do in planting or establishing the rootstock to make the rootstock more drought tolerant later on?
So the rootstock itself.
I'm just wondering about, you know, whether, you know, how you establish the rootstock with watering or cultivation practices or competition, or whether those would change the root structure or architecture to help you much, you know, really later on.
Yeah, so I think for that first year, it's really crucial to get enough root growth. So you don't want to try to like bring up the new trunk or cordons too quickly, because then you won't have enough resources to establish a strong root system. And if they're too, you know, you don't want them to be stressed that first year anyways. Because of that, you want them to be you want them to have enough resources to do like really good below ground growth. So I think like that's, you know, like those practices matter for establishing the rootstock. Yeah, I think like everything else is kind of maybe like standard practices you already do. Like, you know, you want to make sure you don't have hard pan layers that are going to block the rooting depth. And I think that's probably like, yeah, I don't know how much you could, as long as you're getting like healthy growth, healthy vigor, your soil has been appropriately taken care of. I don't know how much control you have over the rootstock at that point. So I think it's mainly avoiding problems more than it is like, oh, this will definitely improve it.
And then letting the genetics take care of it.
Yeah.
Okay, cool.
Well, we're at 1:59. So I don't know if somebody still has a burning question, but I don't.
Oh, one just came in.
Oh,
Just under the wire.
just out of the wire. Okay. Okay, so this is actually more a comment on the previous question.
Mm-hmm.
So before the rootstock is planted, 1 year before the rootstock is planted, it can.
I think what they're maybe asking is, how would your answer to my question be different if you were planting a grafted vine that was already pre-grafted to the scion?
There you go.
Oh,
Because if you're planting rootstock and you're only worried about establishing the roots, it's one thing. But if you have a grafted vine, and
yeah,
you need
so
to take care of the scion part as well, how would
when I was answering, I was picturing kind of it's already been grafted,
Right.
and you're planting something that's already been grafted. So yeah, I think the main thing there is still like you don't want to try to take up like establish the trunk and the cordons like too quickly, because that's really effective at competing for resources from the root system. So I think that same thing would apply to this
still
question
focus
too.
on the rootstock for,
Yeah.
well, at first, yeah, until it's established.
Well, I don't know if Dave wants to, but otherwise I'm going to say, Megan,
You go ahead.
thank you very much. It was very interesting. Every time I listen to viticulturists or plant physiologists, I learn new stuff. I hope everybody else have learned at least something new or were reminded of something they have forgotten about. It was great seeing all of you or seeing your names. And hopefully tune back next week. We're going to be a little bit more winery focused, but you know, we all need to expand our knowledge everywhere. Have a great rest of your week. Hope to see all of you next week. Thank you for Karen and Caroline for organizing everything. Thank you, Megan. Thank you, Dave. Thank you that I only have to arrive. And next time I will drink my coffee before we start.
Thank you so much for having me. Thank you,
Thank
Karen
you.
and Caroline, for organizing, and Dave and Anita for hosting. Thank you all for coming.
Thank you, everyone. Bye.